let's talk about the first two stages of embryonic development. So first of all, we have fertilization, which is defined as the formation of a diploid zygote from a haploid egg and sperm. Fertilization will happen in 3 steps. First of all, sperm must penetrate the egg's protective layer. Secondly, the egg receptors must bind to the sperm. And lastly, egg surface has to change so no polyspermy occurs. Polyspermy is defined as the entry of multiple sperm nuclei into the egg. A couple of notes before we begin. Fertilization in mammals occurs internally. For humans, such as ourselves, we have what we call capacitation. Capacitation is basically having secretions in the female reproductive tract that alter sperm structure and mobility. So afterwards, the sperm cells have to travel through these whole bunch of follicle cells which provide support to the egg before reaching the zona pellucida or pellucida, which is considered to be the extracellular matrix of the egg. And that is where we start our acrosomal reaction. So this is just a general outlook for what an acrosomal reaction will look like. And we'll use the model example of sea urchin to demonstrate our ideas. So first of all, the sperm will meet the coat of the egg. And that's where we begin our acrosomal reaction. Our acrosome, which is a specialized vesicle at the tip of the sperm, will release hydrolytic enzymes. These hydrolytic enzymes will digest the material coating of the egg, and that will allow our acrosomal process, this little sperm structure, to penetrate the coat. The proteins on the tip of this acrosomal process will bind to the egg receptor proteins. This will initiate the fusion of sperm and egg membranes, and afterwards, one nucleus will enter the egg cytoplasm, and the ion channels for the egg membranes will start to open. As these ion channels open, sodium ions will diffuse into the cell. This diffusion is known as depolarization, and that will result in a decrease in the membrane potential. This will prevent additional sperm from fusing with the egg plasma membrane. As a whole, this is considered to be the fast block to polyspermy. Fast because, well, it occurs within 1 to 3 seconds, but it only lasts for about 1 minute. Also, it's not identified for mammals. Anyways, because it only lasts for 1 minute, we have to find a more permanent solution. So that's where we have the cortical reaction. Basically, to start off, we have the cortex, which are vesicles beneath the egg plasma membrane that turn into cortical granules once the sperm binds to the egg. The cortical granules fuse with the egg plasma membrane and release its contents via exocytosis. They'll release their contents below the vitelline layer formed by the extracellular matrix. So that starts our cortical reaction. So the vitelline layer starts to be lifted by these little vesicle materials, and then it hardens to form our fertilization envelope. So this fertilization envelope is considered to be a more permanent solution as opposed to our fast block polyspermy. And this will then be considered to be a slow block to polyspermy. Also, when this occurs, enzymes will start to clip off other receptor proteins, so no other nuclei can bind to them. Now, the formation of this fertilization envelope requires a high amount of calcium. Calcium is needed for egg activation, and egg activation includes cellular respiration increase and protein synthesis increase as well. So it requires a high amount of calcium to have the cortical granules fuse with the egg plasma membrane. So egg activation after the sperm and egg nuclei emerge. So then we have cleavage. Now, cleavage is when we have this rapid cell division after our fertilization, without, of course, cell growth. Basically, during cleavage, cell cycles will only have 2 phases, the S phase in which DNA is synthesized, and the M phase, which is where mitosis happens. For mammals, this occurs between 12 and 36 hours after fertilization. So, in cleavage, rapid cell division, the egg cytoplasm will be partitioned into smaller cells. These smaller cells are called blastomeres. Now, after a whole lot of partitions, 5 to 7 cleavage divisions, we'll have a blastula. A blastula is this hollow ball of cells. Now, it's called hollow because inside is this fluid-filled cavity, what we call a blastocele. Now, it should be noted that for a whole lot of different organisms, we're gonna have a whole lot of different cleavage patterns. And it's mainly because of this distribution of yolk. Yolk is just stored nutrients. And this distribution of yolk will be a key factor into influencing cleavage patterns. So in this case, we're going to look at a frog. Now, in this frog, we're going to have an animal pole and a vegetal pole. The vegetal pole will have a higher yolk concentration, meaning that the animal pole is what's going to be divided. Now, because of this difference in yolk concentration, we're going to have a difference in hemisphere appearance. So, division. For the first 2 cleavage furrows, we're going to have equally sized blastomeres at the top. And for the third one, we're going to have an asymmetric cleavage furrow right there. Also in frogs, we have a gray crescent, which is a structure that curves opposite from where sperm enters. And it's going to mark where its future dorsal side will be. Dorsal side meaning referring to its back. Also, there are also different types of cleavage. There's holoblastic cleavage and meroblastic cleavage. For holoblastic cleavage, it's going to have a complete division of the egg, like this case, and it occurs in animals where there's little to moderate amounts of yolk. This includes sea urchins, frogs, and mammals. There's also meroblastic cleavage, which is incomplete division because there's just so much yolk within these eggs. This will occur for reptiles and birds. Basically, because there's so much yolk, the cleavage furrows won't be able to pass through, so it looks just like a mess. Anyways, cleavage will stop when the ratio of nucleus material to cytoplasm material is large enough. And so, after cleavage is completed, the rate of cell division will start to slow down, and our normal cell cycle will be restored. Now, we want to start with this process of morphogenesis. Morphogenesis is considered to be the process by which cells start to occupy their appropriate locations and the animal body shape and structure starts to occur. But before we get to morphogenesis, we have 2 things that are responsible for it. One is gastrulation, and the other is organogenesis. Organogenesis, a video that we made before, refers to the formation of the organs. And it's actually the 3rd step in embryonic development. The 2nd step is called gastrulation. And gastrulation is defined as having a set of cells at or near the surface of the blastula to move inward to interior positions. And this movement to interior position will start to establish cell layers, also known as germ layers, and this primitive digestive tube. So let's look into gastrulation a little bit. Also, this blastula will start to change into this 2 to 3 layered embryo called a gastrula. And these layers are these germ layers. So, germ layers. We're going to have 2 to 3, depending on what type of organism you're looking at. The outer layer is called the ectoderm, and it's going to consist of things like the epidermis and sensory systems. And the inner layer is called the endoderm, and it'll start to line the embryonic digestive compartment and tract. It also consists of the respiratory system, the excretory system, and the reproductive system. Now, the mesoderm is for trypoblasts, which are organisms with a bilateral symmetry. And it's considered to be the middle layer, and it'll serve for skeletal, muscular, excretory, reproductive functions. So let's go ahead and look into this process of gastrulation for sea urchins. So the process of gastrulation for sea urchins will begin at the vegetal pole, and in this case, mesenchyme cells will start to detach from the blastocele wall. So again, blastocele is that little cavity in the middle, and all of the different germ layers are color-coded. So the mesenchyme cells are basically future mesoderm cells. They start to detach from this wall, and the wall starts at the vegetal pole. Now as they attach, they start to move inward. This inward movement is called invagination, the inward folding, the infolding of sheath cells into the embryo. So the inner part of this infolding is called the archenteron. It's the blind-ended tube. And the outer part is called the blastopore. And it's the open end that will eventually become the anus. Now, as we move further inward, we're going to eventually reach the ectoderm once more. Now, the opening for the ectoderm will become the mouth, and the first opening will become the anus. And from beginning to end, anus to mouth, that's going to be our rudimentary digestive tube. Now, some vocabulary before we end this video. Protostomes have their mouth formed from the first opening, and deuterostomes have their mouth formed from their second opening. So looking at this, we can tell that it's a deuterostome because the mouth comes from the second opening, and the mouth comes from the second opening, and the anus comes from the first opening. Anyways, that's pretty much the process of gastrulation, the inward moving of cells for a sea urchin. Now, there are a whole lot of other types of gastrulation. Gastrulation for frogs, gastrulation for chicks, and gastrulation for humans. Gastrulation for chicks will include the primitive streak, the epiblast, and the hypoblast, and gastrulation for humans will include the blastocyst, also known as the mammalian blastula, intercell mass, trophoblast, and extra embryonic membranes. So we'll get to these other types of gastrulations in other videos, but for right now, remember what fertilization is, remember how it occurs. It's split up into the acrosomal reaction and the cortical reaction. After fertilization, we'll start to have cleavage, and cleavage is the rapid cell division. And after cleavage, we'll start to have gastrulation, where the set of cells start to move inward. And those are the first two, or I suppose you can call it first three stages of embryonic development.